vaccine manufacturing program at PATH. For those that are not familiar, SBM provides technical assistance to LMIC vaccine manufacturers to enhance their abilities to develop and sustainably produce safe, effective, and affordable essential vaccines of international quality to meet global public health program needs. And for those that aren't familiar with PATH, PATH is a global health nonprofit at work in over 70 countries around the world. We make good health easier to assess in low resource settings by creating affordable health products, strengthening health services, and advancing effective health policies. For today and tomorrow, this webinar will be recorded and it will be available on path.org in October, in addition to the presentations that you will be seeing today. Please, during this time, keep your microphone on mute and your camera off so that we don't have disturbances of our presenters. There'll be a few minutes for questions after each presentation and at the end of each daily webinar session. All questions should be posted to the MS Teams meeting chat, where they will be monitored by the meeting moderators. Feel free to enter those questions at any time during the discussions and presentations. And all registered participants will receive an email survey to share your feedback on this webinar. As I said, this is one of a number of webinars that we put on through the SVM program, and we value your feedback and how we can further enhance them moving forward. This is the agenda for the next 2 days. As I said, my name is Tara Tagmeyer, and I'm with PATH. Next up will be Colette Cote with PathoQuest, and she will be discussing NGS process overview. Arifa Khan from the US FDA will provide the current status of NGS for adventitious agents and regulatory expectations. And then she will be followed by Bradley Hassan and Leila Diaz for a case study on NGS validation for adventitious agent detection in a vaccine. So today's focus is really setting the stage of NGS and its application to adventitious agents. Tomorrow is going to be more of a product-specific focus. Since NGS has many applications, we felt that it would be good to provide it for a product agnostic focus with adventitious agents today, and then we'll zoom into the product-specific focus. And with that, I think we are going to jump right into the agenda. And we again will start with Colette Cote, who is the Chief Scientific and Program Officer with PathoQuest. Colette, are you with us?
I am here, and everyone can hear me okay?
Yes, Colette.
We can hear you.
All right. Perfect. And I see you're sharing the slides, so we'll go with that one. So yeah, so thank you to the organizers, as well as thank you to the crowd that has joined us today for this opportunity. My goal is to give you essentially a high-level overview of what next-generation sequencing is, where it came from, how it evolved, how we're applying it today, and really set the stage for the speakers that will come after me. And I'll try to answer a lot of the basic questions that we hear every day about this technology. So with that, if you could, next slide, please. So I'll just take a step back, though, at least for this audience, and just remind everyone, of course, throughout the manufacturing of biologics within our industry, and it really doesn't matter what type of biologic, whether it's a viral vaccine, oncolytic virus, gene therapy vector, so forth, there is a manufacturing process, and throughout that manufacturing process, there are some testing requirements that are required by regulatory agencies and the health authorities. And that could be from the early phase of this with your starting materials, whether it's a cell line, a viral seed, raw materials, throughout the manufacturing and the scale-up expansion process, if there is one, and it may vary a little bit from what's shown on this slide. but throughout purification processes and even to final fill and finish, that there are these testing requirements, and they fall into two basic categories. So one is adventitious agent detection. So that is your detection of contaminating viruses of concern within your product, as well as genetic characterization. Now, throughout this entire process, and it really doesn't matter if you're in a development phase or if you're at a commercial product phase, the testing requirements may be modified a little bit, but these are in general areas where next generation sequencing can be applied as a testing strategy. Next slide, please. If you look at what traditional biosafety testing is, there are a collection of these assays, and they're shown in the middle of this screen. And typically, regardless of product, might be a complex testing package, and that package will depend, this precise package will depend on the product and the regulatory guidance that's needed. These typically are, in these days, multiple aging assays with a lot of animal-based testing or even quasi-animal-based testing. These are traditionally low-resolution tests that give you some indication that something is going on, but not necessarily full resolution. In many cases, these have extended timelines. For example, your in vivo assay, your in vitro assay, 28-day assays, longer months of assays. And they can come at increased costs because of a full collection of assays. The concern with a lot of these assays, and these are legacy assays that have been out for decades now, obviously is resolution. It can be specificity in many cases. There's a lack of test system susceptibility. For example, in vivo or in vitro model systems are only contaminated by viral agents that they're susceptible to, and not every one. And there may be instances of assay interference as well. For example, your product may interfere with the test or prevent the test from being used. In a perfect case for that may be an oncolytic virus, where you might need a neutralizing antibody in order to enable a functional in vitro test and so forth. So there are a number of classical test concerns, and these change as, again, the product changes. However, the goal, of course, is as technologies evolve, is to apply state-of-the-art technologies. Next slide, please. So in our ideal world, we would have a simplified biosafety testing strategy where you have one test that could replace several assays, eliminate animal and quasi-animal based testing. You'll hear more about this in some of the regulatory talks during this conference. The idea is to replace those, to minimize use of those in light of the 3Rs principles, as well as to combine things that are traditionally, for example, PCR tests or in vitro tests or immunoassays, 9CFR tests, and so forth, to really compress the testing package down. Not only do you have less tests to run, less material used, less test sample used, so you can preserve more for your final endpoint product. You can get improved resolution, improved sensitivity, specificity, and so forth. So really, where next generation is becoming well suited for now is to replace many of these tests. And in fact, a lot of the guidance documents have started very explicitly calling out next generation sequencing as a replacement to a collection of these tests. Some of the other advantages for using a technology like next generation sequencing is you can not only you do see, again, the improved resolution, improved specificity or sensitivity of an assay, but you can get simplified matrix interference assessments. You can simplify or even eliminate the need for method adoption. It can be built into these assays. And then there are other advantages, including it's molecular biology, a molecular test. So there really is no need for neutralizing antibodies, and it may even eliminate other pre-work cell cultures and so forth that may be required for other tests that next generation sequencing does not require. So with that, again, just wanted to set the stage as where NGS is starting to fit into biosafety testing. Next slide, please. So first, another interesting facet here is I wanted to give a little bit of a historical foray into sequencing, and the reason for that is there's a lot of folks out there who think next generation sequencing is brand new, and it really isn't. So if you look back throughout the time here, and this is a nice historical slide, if you remember, Sanger sequencing and that dideoxy sequencing really came out in the mid 1970s. So this might predate a lot of people who are on this call, and that was a steady state technology for decades, really up until the late 1990s. And then newer technologies started to come about that included technologies that started to resemble more of the next generation sequencing technologies that we see today. So from about the early 2000s until today is really the era of next generation sequencing or high throughput sequencing or massively parallel sequencing. There's a lot of different names for these. Massively parallel sequencing was probably the first moniker for it. Next generation sequencing really now is specific for a type of sequencing, so it's not used as much in that context. But high throughput sequencing, so how do you go from a single molecule at a time sequencing to billions of molecules, DNA molecules being sequenced at the same time. So really this started in the what we consider next generation sequencing today, really started in the early 2000s on those platforms. And it was only around 2008 when it's really started to enter the biosafety testing space. Prior to that, and even to this date now, a lot of the focus is on whole genome sequencing of humans, but it was hijacked in the mid 2000s really to meet the needs of this industry. And of course, the whole point of this is to use this platform and find cost-effective ways of generating a lot of data for different applications. Since the early 2000s to today, a lot of the platforms have changed, and I'll speak about some of the main platforms that are used in the industry today, but know that there's a lot of them out there. The platforms have changed, the chemistries have changed, the amount of data, the type of data has changed, but really the more interesting thing that has changed is the guidance documents and the acceptance of this technology now in your basic portfolio of testing strategies. So using it as a tool for testing strategies. So today in the guidance documents, particularly the ones that have been changed in the last few years, you will explicitly see the language next generation sequencing, high throughput sequencing in there as testing strategies. So again, it's about a 20-year-old technology these days, but it's really only getting its traction now. Next slide, please. So what are the differences between these sequencing systems? You hear the words first generation, second generation, and third generation. So first generation really is your basic Sanger sequencing. And this is the generation that existed from the mid-1970s until probably the late 1990s. This is probably your best understood instruments for this are the ABI platforms, so the 3730s, the 3500s, and so forth. This is called sequencing by synthesis, and I'll go more into the details of this. The sequencing by synthesis or short read sequencing. And with that, I mean, the length of sequence that you can generate out of this is typically, 1 kilobase or less. And it can be on a smaller scale. You're getting, if we think about Sanger sequencing for testing strategies, you're thinking 4x bidirectional coverage. It's very, very low output, very low yield. Low resolution on that. And again, so second generation sequencing was a dramatic change from Sanger sequencing, and this is where the massively parallel sequencing came in. And some of the earlier systems like the Solexis system, the Roche 454 system, those are systems that have long been obsolete, but now even the Illumina system is the most current version of it. This is also a sequencing by synthesis type of instrument, and a short read sequencing instrument. In fact, it's actually shorter in many contexts than Sanger sequencing. But it changed the scale of sequencing. So no longer do you just get, the output for one single sequencing DNA molecule, but you get it from millions of to billions of DNA molecules at the same time. Along the way, a different generation came out. This is third generation, and there's a slight correction to make on this slide, but I'll get to that in a moment. And this is really illustrated by sequencing systems like Oxford Nanopores, collection of sequencing system, or PacBios. And this is the actual technologies vary a little bit. Nanopore sequencing is probably the way it's most commonly thought about. That's really the Oxford Nanopore system. And again, I'll go through the details of that. The PacBio system uses a slightly different version of that called SMART, single molecule real-time sequencing. What this is, the third generation is considered long read sequencing or all read sequencing. And the major difference is length of each individual molecule. And there are many applications where you really do want long reads. And by long, I say up to about 200 KB is the current kind of maximum that we see for this platform. So really you see the evolution from small scale short reads to larger scale long reads. And again, right now we see a lot of activity in the second and third generation sequencing space in new chemistries and applications for that. And I'll go over a little bit of that. So just think of next generation sequencing. I always call it as Sanger sequencing on steroids. It's an advanced technology enabling analysis of millions to billions of individual DNA molecules rapidly and simultaneously. And the key there is individual DNA molecules. You get information about every single DNA, and it's unique information that you can use as unique information. Sanger is considered low throughput consensus sequencing. You just get an average of sequences. The other major difference in these platforms is NGS is sequence agnostic. In other words, you don't need to know anything about what you're trying to sequence, but you can still sequence it by the use of these adapter sequences and other tips and tricks whereas in Sanger sequencing is primer dependent. So you really do need to know some sequencing in order to prime that sequencing reaction. So there are some significant differences in these platforms. And again, I will speak to the primary ones that are used in our space these days, which is Illumina and Oxford Nanopore, although keep in mind there are other platforms available. Next slide, please. So I'll go a little bit through the chemistry here, not to bore anyone, but just to explain it a little bit. So again, on the left-hand side is first generation sequencing. So this is your Sanger sequencing, and most people are familiar with this. This is termination sequencing. So you, and it's sequencing by synthesis. So you're actually synthesizing the complementary strand of the DNA molecule of interest. It's primed using a primer, a short oligo sequence. And the way that the mechanism works is there's an incorporation of the next nucleotide that is complementary to the target sequence. And those nucleotides have specific dyes on them that are specific to each base. And there are some terminating ones in that mixture, low level of terminating dyes. So basically you can identify every position on that molecule. And it's resolved through capillary electrophoresis. And you have to do individual reactions for forward and reverse strand reading. And it's relatively low throughput. And what you get is you can see the little trace, the electrophoregram trace on the bottom there, that shows with the different peaks what the different bases are. Now, if you get a mixed base, you'll see a mixed peak. And so there are some limits in resolution for Sanger-based sequencing. So typically, you can't resolve below 20-25% mixed in the population. So there are some limitations for that. If you move to second generation and Illumina sequencing, it's very similar to Sanger-based sequencing. So it also uses, it's also sequencing by synthesis. So you have your strand that you're then adding bases, your nucleotides to as you flow them over this flow cell that are color coded. And different chemistries use a different number of colors for this, but the instrument is designed to recognize the expression of that color in there. The difference here, of course, is you have a strand, so you have your DNA strand. And really, you're adding these adapters to the ends of them. These are universal adapters. So you have your sequencing strand, your end adapters, and those adapters are used as your priming site for sequencing. So you make what's called a library. So a library is just a complex mixture of different sequences. This library for the Illumina platform must be double-stranded DNA. So you must convert anything RNA, single-stranded DNA to double-stranded DNA in order to sequence it. We use these adapters. These adapters have other sequences on them that allow them to stick to the flow cell, which is what you see those little loops in that middle panel of the middle image, and it gets amplified on this, basically on the flow cell. So you have many copies of the same sequence within a general area, and then the chemistry flows over those, and as it adds a base, again, there will be this emission of light, and that will get detected by the system. And because it's doing it over a large footprint, it's actually collecting information on individual molecules. So unlike Sanger sequencing, which just kind of sees all molecules at the same time, but it makes a consensus sequence out of that, second generation sequencing will actually resolve individual DNA strands. And those strands are barcoded. They have unique identifiers so that you can multiplex samples and you can do a lot more data analysis on those. Now, these systems, the Illumina system, again, is a short read sequencing chemistry. Well, it has very high throughput. Depending on the instrument, it can take a lot longer to sequence. It could take, sequencing runs can be done within hours or they can be done within days, just depending on which platform that you're using in that for that particular vendor. However, the nice thing is because you generate, you know, just massive amounts of data, there's this great ability to multiplex it. So you can mix samples that would have individual barcodes added during this library construction process. And I'll go through that in a little bit. But third generation sequencing is very different. It does sequence individual molecules, but it does it in a very discrete way. So what I show here is actually Oxford nanopore sequencing technology. And it uses these nanopores. So these are physical biological nanopores that act as a ratcheting system where a DNA strand will go through the pore. And as the DNA ratchets its way through the pore, or it can be RNA in this case, so the ONT system is capable of direct sequencing of RNA or DNA. As a molecule moves through the pore, each base has a specific current that ionic current that it creates, and that is recognized by the sequencing system. So it doesn't have to use a color-based system. It's actually using a current-based system. And it just ratchets through and it reads every base and it collects that information that way. And by the nature of these nanopore systems, you can achieve long or even ultra long sequence reads these days, which will provide a lot of data. Now, it's a medium throughput compared to, for example, an Illumina system where you can get 1 billion reads on an Illumina system. With an Oxford nanopore system or some of the third generation systems, the data output, it isn't as large, but you get a lot of value in the data because of its long reads. So that's just a very high level overview of the different generations of sequencing. Now we're waiting to see what fourth generation sequencing will look like, again, with the platforms that are out there. Next slide, please. So if we go really simplify next generation sequencing down, there are 3 basic steps. First step is sample processing. That is just extracting the nucleic acids from a test sample. So any test sample that is assumed to contain nucleic acids is a perfect test sample for next generation sequencing. And it can even be fragmented nucleic acids, again, because short read systems don't really care about that. So the first process is to extract nucleic acids, and then you go into this library preparation process, and that is really just converting those nucleic acids into a sequenceable molecule. And it's platform specific. Again, as I mentioned, for Illumina, these can be double-stranded DNA. They need to be double-stranded DNA molecules that have these adapters and barcodes that are then added to the ends of them. The next step, of course, is sequencing of that library. And that's loading your sample onto the sequencer and doing the chemistries that are involved in that. And of course, the last step is data analytics, bioinformatics, and there's some sub-steps in there about migrating your data and where you're going to pre-process, how you're going to pre-process it as other considerations. But those are the 3 main steps. But a few things to keep in mind too here is, for the critical quality, critical assay attributes, obviously higher quality DNA or nucleic acid that goes into the system means higher quality data. And you want to be able to apply smart bioinformatics to that, meaning exactly what are you looking for? Ensure that your library is going to generate that information for you before you start. So if you want to look at RNA, you want to start with purified RNA. If you're looking at just DNA or genomic DNA, obviously you won't have the same type of library prepared for that. In our world, we want these to be fully validated end-to-end GMP workflows, and you'll hear more about this later today. And throughout this process, of course, it's critical to have intermediate quality control steps. These are go, no-go gates. This is a longer process for sequencing in general is a longer process. There's more time involved. There are more steps involved than, for example, Sanger sequencing or even PCR-based methodologies. And it can be in certain platforms a little more expensive to do, so you don't want to get to the end and find out something went wrong. So we introduce these quality control steps, as well as there's a criticality for assay controls, the nature of those controls. And again, you'll hear more about this later today. And then from a bioinformatics point of view, unlike Sanger sequencing, which you can just process data on any standard laptop these days, with next generation sequencing, the volume of data is significant, and therefore you want to make sure you are in the appropriate computing environment. And typically now these are cloud-based computing environments, including computing as well as storage and archiving areas. You want these to be scalable, dynamic environments that can accommodate changes in sizes of the data, um, as well as apply the appropriate bioinformatics pipelines to meet whatever the need is, whether it's a regulatory need or your own expectation. And these may require use of databases or data banks of data for analysis, particularly for adventitious agent testing. And there may be other needs for secondary qualification, for example, for adventitious agent testing to remove false positives in the data output and so forth. And these pipelines are typically tailored very specifically for applications, and they can take a bit of time to run just depending on the nature of that pipeline. If we move to the next slide, please. So if we dive a little bit deeper into the Illumina workflow, here, again, as I mentioned, starting materials can be anything. It can be anything that it's assumed to have nucleic acids as long as you can recover those nucleic acids. In our world, this typically is cell banks, virus seed stocks, and other reagents, raw materials and so forth. Once you extract the nucleic acids, there may be some other pre-processing steps. For example, there may be need for ultracentrifugation or nuclease treatment, or just depending on exactly what you're looking for, and ensuring that you're extracting the appropriate nucleic acids. Is it total RNA that you want? Is it genomic DNA, or is it a combination of that? Once you have your nucleic acids, you want to do some QC checks on that. And that is just to make sure that you have sufficient material to proceed to the library construction, or that you have the appropriate quality of material to proceed. Is it intact or is it fragmented and so forth? Once you have that nucleic acid extracted, QC checked, your next step is, of course, library preparation. And I went through the process kind of briefly, but really, again, this is conversion of the starting material to double-stranded DNA. So the addition of these adapter sequences that may contain barcode sequences. Again, that allows clear sample identification. So you can do sample-specific barcoding. You can mix samples when you sequence it for more efficiencies. And then you can demultiplex your sample so you're only analyzing a specific barcoded data set. These libraries are then added to the flow cell with the chemistries. We go through something called cluster generation, which is where it amplifies the individual molecules once they're attached to the flow cell to give sufficient DNA coverage for the sequencing process here. The next step, of course, actually before that, there is some QC steps. So you want to make sure your library has the right profile, the right yield, right quality before you put it onto the sequencer. It can then go onto the sequencing instrument, and there is a series of different sequencing, particularly in the Illumina series. Smaller scale instruments like iSeqs and MiniSeqs and MySeqs, to larger scale instruments like NextSeqs and NovaSeqs, with NovaSeq currently being the largest instrument, at least in the Illumina series, where you can generate hundreds of whole genome sequencing data sets if needed. Most in this industry use a MySeq or a NextSeq sized instrument, and you can still multiplex a significant number of sample on those instruments. After the sequencing run itself will provide a number of quality control checks that are then used to ensure that your data is of high quality for analysis, and then you can go into the appropriate data analysis workflows in whichever environment you may be working in, whether it's an AWS cloud, Google, Azure, or anything like that, or even on-premise analysis. And the analysis here can be just tailored, again, to the application. Next slide, please. The Oxford Nanopore workflow or long read workflow is very, very similar. A lot of the ends, if you think of the book ends, are pretty similar. The library preparation process is a little bit different here. It can also be barcoded and so forth. But these again, this workflow is tailored for longer reads, so high molecular weight DNA is key for this, for these applications, so that it may change your extraction steps or some of the pre-processing steps. Again, the for this Oxford Nanopore system, you can do direct sequencing of RNA or DNA. Base calling here is done in real time. So as it sees it, it reads it and will generate that information. Unlike an Illumina system, which actually generates images that has to then get converted to base information. There's also some QC steps here. Again, they vary a little bit because of the instrument, but you can use a lot of the same pipelines and tools, but they do have to be tailored to adjust for long reads as opposed to short reads. So next step, please. Slide, please. Again, just high-level data analysis. You're going to get gigabytes, sometimes terabytes of data generated. That's a significant amount. These are typically FASTQ or system-specific files. You want to provide some data integrity checks on there, and you may need to do some demultiplexing before you can look at the data. Typically in these workflows, there is a first step called quality trimming or pre-processing, where you want to remove poor quality reads. So you have these quality scores for every base position and every read. You may want to trim the ends to get rid of the adapters. They're not very useful in data analysis. And there may be steps where you want to remove other types of sequences. You want to subtract these sequences from the data set because they're not meaningful to your analysis. And then you can go into your data processing workflow. And again, it just depends on what the need of that workflow is. It for adventitious agent detection or identity detection and so forth. So it can vary a little bit. Next slide, please. Well, I know we get some questions about equipment and infrastructure considerations. I'll go through this very quickly here, though. In order to implement next generation sequencing in any lab, you need standard molecular biology equipment for the most part. So your basic stuff, your vortex mixers, your pipetters, your centrifuges, thermocyclers, and so forth. Some electrophoresis equipment, a spectrophotometer, fluorometer, and so forth. There may be needs for some workflow specific equipment like ultrasonicators or ultracentrifuges. But then, of course, there's the nice to haves. If you're at high scale, which in this field isn't necessarily large scale, high capacity sequencing, you can also take advantage of automation. Liquid handlers, either for extraction or even the library preparation. And there's a lot of brands that are out there. I just highlight some. So this is just, if you're adept at molecular biology, you can make a great NGSer. You may also want to think about equipment for quality control assessments. For example, you want to do purity checks, concentration checks, integrity checks. You would use your standard equipment for that. And the most common ones here are like the qubits, the nanodrops, SpectraMaxes, bioanalyzers, and so forth. So there isn't a whole lot of specialty equipment maybe outside of your sequencer itself, but things to keep in mind is your throughput and scale. What are your read lengths that you want? What scale do you want? Do you need a lot of data? Not so much data. Be mindful of equipment obsolescence. This is a fast-moving field that's still in the RUO space, which can mean that some equipment can be made obsolete sooner than you would want it to be. But there are a lot of stable platforms out there, Illumina being one, Oxford another, PacBio, and so forth. Of course, in our world, we are interested in GMP. So there is a need for 21 CFR Part 11 or Annex 11 compliance, and it can be a little bit challenging sometimes when all of your equipment is designated as research use only. So there may be other requirements there to ensure that your equipment can be validated, and you may hear more about this later today, as well as think about integration into other systems like LIMS systems and so forth. From a sequencing perspective, your considerations there is, do I need long reads, all read lengths, or short reads? Lots of different platforms can meet those individual needs. Some are more well known for different types of applications. For example, the ABI series, you tend to think of amplicon-based sequencing there versus, for example, in Illumina, where people think of it as a little bit more broad application. Considerations there is operating system. Sometimes people get surprised by Linux systems. They do provide an interesting challenge. Throughput and scale, do you need a lot of data? Not so much. Again, what is the read length requirements? Are you doing integration site analysis? Requires long read lengths. If you're just doing an identity test, not so much. You can get away with short read lengths. What is the range of desired applications and supporting chemistries? There may be a need for an amplicon panel in 1 minute, and you want to do an identity test in the next, and adventitious agent detection in the next. You want a platform that can accommodate all of those. Keep that in mind. I already mentioned equipment obsolescence. I already mentioned 21 CFR Part 11 compliance requirements, but also vendor lifecycle management and software upgrades. A lot of systems, particularly O&T, give you the option to upgrade your operating system or your software very frequently, which in a GMP environment you have to be careful about. And then for data analysis, you have your computing environment and supporting infrastructure. Are you going to be cloud-based or on-premise? Either one can be set up pretty easily, but there are some advantages to cloud-based because of scalability. You can do in parallel processing. You don't have to worry so much about queuing up samples for analysis. You can actually in parallel analyze those. Where are you going to store and archive your data? These can be large data files depending on what you consider raw data. They're the image files, FASTQ files, and so forth. What's the pathways that I need to take to get to these environments? In ensuring data integrity and security checks. So the agencies are very savvy about that. What does my pipeline look like? Is it going to be open source? Is it going to be customized in-house? Proprietary tools, exactly what am I using? Same thing with databases and data banks. Are these easily accessible through in the public realm, or is it something that I have to develop here? And then, of course, with data analysis, the keys are really 21 CFR Part 11, Annex 11 compliance requirements, access rights, version controls, electronic signatures, data integrity checks, and so forth. So there's a lot more emphasis on data security and integrity at that part. Next slide, please. Just other considerations, and again, you'll hear more about this, workflow controls. What are the nature of the controls? Are they in parallel controls, spiked in controls? And there's different types. You can use infected cells, intact viruses, nucleic acids. Questions you might ask yourself, what's the long-term availability of these controls? Are they publicly accessible? What's the level of characterization and available documentation? Remember, next generation sequencing will reveal everything about your sample that may not have been revealed through the vendor themselves. They may not have done as deep a characterization. And you have lot-to-lot consistency because that can actually change the nature of your assay. And then, of course, quality control assessments. How many controls are needed? Where do you put them? What's the nature of that assessment? Is it quantitative? Is it qualitative? And I've mentioned some of these, for example, concentration assessments, purity assessments, integrity assessments, fragment sizes, data quality, and so forth. These are all key considerations. Again, they're application specific, so I won't go into those very deeply. Next slide, please. So from an NGS point of view, how do you simplify testing for adventitious agent testing? Well, there are two main types of assays in our industry these days, and they're all based on one simple question: are intact cells present? And the reason that question is asked is because the assays will actually target different features of a sample for adventitious agent testing. So if cells are present, if you're trying to screen a master cell bank, end of production cells, the legacy assay in the field, right, is a transcriptome assay or transcriptomic assay. And this is an RNA-based assay, and I'll give a high-level overview of it. Used to detect replicating viruses. And again, this is the legacy. This is probably the oldest test, at least in our industry. On the other side of that, if you have a cell-free system where you don't have if you're looking for if there's a replicating virus and you don't have cells to analyze it with, you might want to focus on viral particles and so forth. So there's a different assay. It's called a virome assay, viromic assay. It can be used for supernatants, drug product, drug substance, clarified bulk harvest, and so forth. Again, it just targets different nature of material. So in this case, it would be viral particles to look for example, replicating viruses. In either case, these should be simplified 3 Rs compliant testing strategies, and there are a number of them out there from different vendors, including Pathogas. Rapid turnaround times, broad detection capabilities, very different than current methodologies. Conduct all types of replicating viruses with sensitivities that are comparable or better than PCR, which is now seemed as the gold standard. And there's a lot of published head-to-head comparability data in the field right now from different entities, including consortiums, where you can show that comparability to really meet the regulatory requirement for that. So next slide, please. So just as an example, high level, so on the right-hand side is a little bit more details, but as an example of a transcriptomic assay again, so this would be looking for adventitious agents in a cell-based system. And here your test sample is an intact cell, and you may include positive and negative controls, cells that are not infected, cells that are infected, and there may be other controls that are spiked into the sample, for example, synthetic RNAs that can be used to monitor recovery or matrix effects. Throughout this assay, and again, it's very similar across providers, but the goal is to extract RNA, convert that RNA to double-stranded DNA, do some quality control checks, sequence it, typically on an Illumina system, but it can be on even a long-read system. And then once you have that data, compare it to viral data banks and try to ask the question, do I have any viral sequences in my data set? And if so, use some secondary qualification steps to really confirm if they are truly a virus or not, or to confirm if they are truly a replicating virus, which is the more important feature of this. Just detecting a sequence isn't enough. You have to be able to put context around that sequence, and really that is to see if it's replicating or not. The nice thing about this assay across the board is it's very fast. You can typically do this assay. The general turnaround times are within 2 to 5 weeks. So if you're comparing it to a 28-day assay, much faster, much more sensitive than these assays, preserves more sample. So there's lower sample inputs, so preserves more of that for your end use. And you can tailor this assay to just depending on what your data analysis system is. You can look at targeted analysis, or you can do an agnostic analysis. I can detect just this small panel of RNAs, the 9CFR panels, for example, bovine panel, HapMap Rap series, or I could look for anything and everything and really do an agnostic analysis. So it has a lot of flexibility in this assay that can be used not only to meet the needs of adventitious agent detection, but you also have a very rich data set that can be used for other applications without having to do any additional lab work. If we move quickly to the next slide. So the converse of this, which has a lot of the same features, is this viromic assay. So again, this is an assay for cell-free materials, and it's called different names across the industry, but in essence, these are supernatants, virus stocks, virus seed stocks, bulk harvest, clarified bulk harvest, in which really what you're looking for there is viral particles, what's in those particles, because those would be indicative of a replicating virus. Some entities use ultracentrifugation to collect those, others not. But the goal is to collect RNA and DNA within those particles, prepare libraries from that material, and you can do it either separate RNA and DNA or combined, sequence it on a sequencing system, and again, perform the same type of data analysis as the transcriptome assay. Again, this is fast turnaround times. It's a little bit longer than transcriptome assay because there's more handling steps, but it's still significantly shorter than a 28-day assay. And a lot of the other features that I mentioned also apply here. And same with the transcriptome assay. The sensitivity of these assays these days are on par with, comparable to, or even better than PCR assays, and certainly better than many of the in vivo assays and in vitro assays for sure. And then there can be use of spiking. So you can spike in certain control viruses to track the performance of the assay, to track matrix effects and inhibition effects. And again, you'll hear more about this later today. So I'll just give that a high-level overview. Next slide, please. If you think, I talked a lot about applying this for adventitious agent testing, but you can think about how do you do a lot of testing with a single assay? How do you really multiplex your testing strategy? And I'll just use an example here for vaccine development, and this would be an AAV manufacturing production. Not only could you do adventitious agent detection in these test samples, and you would simply subtract out the sequence you expect to see, the viral sequence. But you can also do other testing throughout the process. For example, identity testing, identity confirmation of engineered plasmid sequences, or even of that product itself. That could be done with short read sequencing. You can look at replication competency, typically done with long read sequencing, but there's a lot of variation that can be done. Am I getting my are the GAG-PAL or REV or other sequences, just depending on what your vector system is, E1A, E1B for adenovirus, are they somehow recombining and becoming a problematic molecule? You could also identify residuals in capsids. If it's not AAV, what is it? Or if it is AAV, is it intact or not intact? Is it truncated in some way? So you can look at integrity assessments and sequence identity assessments on an individual basis with very high exquisite resolution. So for example, if you have a low level, a rare or ultra rare variant, and I mean something that's within less than 5% or 5% of the population, you can easily detect it on an on molecule per molecule scale with next-gen sequencing. And a lot of these assays can be combined into a single sequencing run, as well as just analyzing the data differently. Next slide, please. Here you can also think of next-gen sequencing for genetic characterization, and of which I just mentioned some, as simplified testing. Again, rapid TATs, limited material use, exquisite sensitivity. You can do individual molecule analysis, and I already mentioned some things about the identity testing here. You can do end-to-end characterization in a single run. You don't have to walk across a molecule. Exquisite variant detection, significantly different than Sanger sequencing. So you can look at subpopulations, other purity determinations. Again, from an integrity assessment side of things, which is on the right-hand side, evaluate intact versus truncated copies. What's encapsulated? Is it host genome or is it an actual AAV and so forth? And do secondary identity confirmation. So what I hope you've heard today, and I'm sorry if I'm speaking really fast, but is there is a broad application for next generation sequencing in our field. While it has very specific applications for adventitious agent testing, and that's probably the legacy assay for next gen sequencing in the field, there's other applications and they're evolving daily. We get new requests all the time about being able to use NGS to solve a problem that a traditional test cannot solve. And hopefully you'll hear more about these applications throughout this conference. And with that, I believe this is my last slide. I think the last slide is, yes. So I want to thank everyone for your attention on this part, and I will be open for questions.
Thank you so much, Colette, for that very insightful overview. At this point, we have about 10 minutes for questions, if there is any that have been submitted to the chat, or if there's any that you'd like to ask now.
So Tara, I don't see any questions in the chat. There is one coming from Indonesia. What platform could be utilized by both manufacturers and NRAs to ensure robust data security and integrity, while also providing the NRA with secure and timely access to analyze NGS results generated by manufacturers?
So that's a great question. As I mentioned, there are a lot of platforms out there. The most common ones within our industry and those which have more of the security, and by that I simply say they've just been legacy platforms or vendors in the field, are Illumina for one, Oxford Nanopore for another, and PacBio. I mean ABI to some extent as well. These are instruments that have been in the clinical space, particularly for ABI and Illumina units, so they have more security requirements around them. I mean, you can get diagnostic versions of instruments versus RUO, research use only versions of instruments. The exact platform that you want to focus on is going to vary by your application. So right now, the platforms that have, and there's a trade-off. So for example, in Illumina series, they're shorter reads. You can, you're going to max out at 300 base reads for most of the current chemistries. What type of information can you get in a 300 base read versus a 1 KB, 5 KB, or 10 KB or plus read? You have to weigh that application. You get a lot of data. You can get billions of individual data points, individual molecule information with a platform like Illumina. On the other hand, as I mentioned, platforms like Oxford Nanopore Systems or PacBio, those are nice because you can do any read sequencing. So from short reads, a few hundred bases or less, up to 200 KB or so. However, the scale of data that you get out of those systems is significantly less than an Illumina series. So you just have to weigh, do I need length more? Do I need total volume of data more? And what's my application? I'm very familiar with Illumina Oxford Nanopore systems. These are systems vendors have moved closer to the 21 CFR compliant space, Part 11 compliant space, Annex 11 compliant. So if you're looking for access points, ensuring your data is secure and so forth, there are platforms you can work with the vendors and get their CSV packages if available, or you can even develop your own with them. There's a lot of utility in that right now, and the agencies are very well aware of this, and you'll hear this later today. The agencies have been following this space for a long time. They recognize a lot of these platforms are still for research use only, and we have had to do workarounds in order to get things established within the field, including working with cloud-based providers for data analysis and access points to data. So with that, I would say we are in a very good space right now. There can still be some improvements, but really the key is more of what platform do you need for your specific application? So great question. So thank you.
Yeah, thanks, yes. Colette. There's more.
Okay.
There's another one for adventitious agent testing. Is there any limitation in the number of viruses that can be detected?
It's as good as your pipeline is or the data bank. So the nice thing about most of the analysis that's performed today, and again, it's very similar across vendors or service providers. We use either in-house data banks, so the databases of viral sequences that are curated from, NCBI and those areas, are even other databases like the RVDB, which is a viral database that's well known in the field. So we use those as the comparator, and then we compare the data set against what's in those databases. And the nice thing about next generation sequencing is you don't need a perfect match when you do this analysis. It's like a blast analysis where you don't need a perfect match, and you can still identify not only closely related species to something that you're looking for, but also distant species. And in fact, we've seen in the field that you can detect things that have very low homology to known sequences. I think there's the interesting case of the rhabdovirus and SF9 cell lines from Arifa Khan's lab is a perfect example of that. But you can see sequences that are, 50% have 50% or less homology to a known sequence, and you can still have the ability to identify it. So there's no restriction, particularly if you're doing an agnostic analysis. If you want to do a targeted analysis, you might, limit your view a little bit, but it doesn't mean that you can't detect these other sequences. You certainly can. So thank you.
And thanks, Colette. A few other questions.
Sure.
There is one on the recombinant yeast strain. They have and want to count the copy number of the inserted gene. Is that possible to use the Oxford nanotechnology?
Yes. Yep.
So that was, can I get the exact copy number of the indicated gene, or just an estimation of the calculation? Is that possible to detect mutation in every copy number?
So this is a multi-answer question. So yes, you can do integration site analysis testing with long-read platforms like Oxford Nanopore platform. The goal, of course, is to sequence from your host sequence through the integrated vector sequence and back into your host sequence. In yeast, it's typically very easy to do because those sequences are limited, and you have to just be able to span that. But you can also do it for CHO systems, for human systems, mouse, you name it. There are methodologies available in the field right now to enable that. I would say CHO is probably the problematic system simply because its genome likes to rearrange a lot. But when it comes to things like yeast, especially microorganisms, much simpler to analyze using a long-read sequencing platform. And there are solutions in the field right now. And it can certainly reach out separately, and we can have further discussions on that application. It wasn't discussed here, but yeah, it certainly is a up-and-coming application.
Okay, thanks, Colette. Couple of other questions. How to check the sensitivity limit of detection of assay for method validation for adventitious agent testing?
So there's different ways of doing that, and I touched upon it a little bit in my talk. When you're looking at something like a transcriptome-based assay, You can spike in synthetic sequences, synthetic RNAs that mimic a lot of these sequences. And there, in fact, there are well-characterized control sequences out there that can be used for this application. They can get spiked in at different areas, and you can monitor the recovery of those sequences. You can also do a bit more head-to-head. In other words, you can spike in, or you can even use infected cells and do PCR comparative outputs for that to look at detection. So from a transcriptome perspective, it's actually pretty straightforward, and those controls can be built into the assay. For a virome-based study, so a cell-free sample, the most common way of looking at that is to spike in virus controls. And there is a collection out there, well-known collection out there, the WHO collection currently of 7 controls, plus I think there's a couple hangers on in terms of minor contamination. So it's really more of a collection of 9 that you can spike in at different genome copy levels within your sample at various steps. And you can assess recovery of that in your data set and evaluate that. And you can even do some head-to-head comparisons using that strategy. So thank you for that.
Yeah, thanks, Colette. There are a couple of others. I think this is covered under your other questions, but we will have to move on with the next speaker. So I give back to Tara for the next one.
Thanks.
Great. Thank you, Katub. Thank you, Colette. And whatever questions aren't answered during the webinar today, we'll document them and ask the presenters to address them in an offline document that we can provide later with the presentation. So for our next presenter, it is Dr. Arifa Khan, who is the head of Molecular Retrovirology Unit, a division of viral products within the Office of Vaccine Research and Review in CBER, which is part of the US FDA. And she will be talking about the current status of NGS for adventitious agents and regulatory expectations. So Arifa, I will stop sharing and hand my screen to you.
Great. Thank you very much. Can you see my slides and can you hear me?
Yes, we can hear you.
Yes, we
and
can.
see the slide.
Okay, wonderful. Well, it's a pleasure to be here to present on our perspective for using our, meaning the OVRR perspective in the FDA, for using next generation sequencing for adventitious virus testing and our expectations. I will first start with my slide indicating that the presentation is an informal communication and represents my own best judgment, and the material in this presentation and my comments do not bind or obligate the FDA. My talk will cover an introduction of NGS for viral adventitious agent detection in biologics. Hopefully it will not overlap too much with what Colette just presented. Then I'll go into CBER's efforts towards NGS implementation, and then the regulatory landscape in the Office of Vaccines regarding NGS applications, and then the acceptance of NGS in global regulatory guidelines. And as I mentioned, my talk will mainly focus on the expectations from our Office of Vaccines, but the information can be extended to other product categories as well as applicable. So I just want to first start with the formal or the accepted definition of an adventitious agent. I'm sure majority are already aware, but I just wanted to indicate it so we're all on the same page. So an adventitious agent is defined as a microorganism that is unintended to be in the biological product. And as you can see, this includes a variety of agents, and the focus of our discussion related to NGS today is viruses. So this just briefly mentions the routine test for adventitious agents. You have testing for non-viral agents for bacteria, fungi, mycoplasma, and mycobacteria. And then you have testing for viral agents. And these include general assays, in vivo assays, including different animal species. You have in vitro cell culture assays, which at least has cell lines from 3 species, one being the same as your cell substrate. And then you have transmission electron microscopy for characterization of your cell bank. And then the reverse transcriptase assay, which currently the one used is the highly sensitive PCR-based or PCR-enhanced RT assay. Additionally, based upon the potential exposure of your product during the manufacturing to other possible materials from other species, for example, trypsin from porcine, a porcine source, as well as serum from bovine or other materials. So there you have the in vitro test for animal viruses, which in the U.S. It follows the 9 CFR 113 Code of Federal Regulations. Then also, if your material, if there's any exposure during the manufacturing or suspected exposure of any raw materials to rodent viruses, then you do the in vivo antibody production assay for mouse, hamster, and rat antibodies and production. And then also, if you have knowledge of any known viruses that could have been introduced, then you can specifically use PCR assays or infectivity assays. Additionally, for viral adventitious agents, especially in our Office of Vaccines, we have a case-by-case recommendations as well. For example, if there's a novel cell substrate, if there's an unknown passage history, then you can also use extended PCR assays, as well as expand your cell culture assays, adding more target cells. And then if there's concern for the presence of latent viruses, such as endogenous retroviruses, or other viruses or DNA or RNA latent viruses, as well as episomal, then you can use chemical induction assays to activate those sequences, and then the detection will depend on the available assays. So, so clearly, these are the currently used assays or the recommended assays, and of course, the recommendations now also have expanded to include some of the NGS platforms, as you'll hear later, and you've already heard before. So I just wanted to mention that the conventional assays, they do have strengths, and they have been generally effective in demonstrating the absence of adventitious viruses for product safety. The in vitro assays, they actually can be highly sensitive for adventitious virus detection, and large volumes can be used. This does have, if you use the extended 28-day full assay for in vitro culturing. The in vivo assays, again, they may be useful for detecting natural isolates, where the host, the animal species may be more sensitive to those strains, but these also have long observation periods indicated here, 18 to 28 days. The PCR assays can be sensitive for specific virus detection, and these can be used for viruses known to occur in the host species of the cell substrate origin, or if there are viruses that may be present in the animal-derived reagents, such as serum or trypsin. However, as you've already heard also, that there are limitations to these assays. Both the cell culture assays and the animal-based assays, they depend upon the susceptibility of the target cells or the animal species to the viruses that you're trying to detect. So it has to be a good match between the adventitious virus and the target cells or species for it to be detected in a sensitive manner. Additionally, both of these assays depend upon replication of the adventitious virus in these in the detection assays, and for the cell cultures such as CPE or hemadesorption hemagglutination, as well as in the animal assays, you have some measured clinical outcome, clinical signs. So here you can only detect, you have limited detection abilities, but they can be sensitive if it's a good match, but they do need to be replicating. But in both cases, you can get interference due to the vaccine virus or the vector virus that may be present at a high titer in your sample. Both of these assays can be interfered. There could be non-specific effects that can be misinterpreted as being real signals. Or you may not be able to conduct the assay at all. For example, if there is lytic effect in the cells that are your target cells. In the molecular assays, they are designed based upon available known virus sequences. So that's a limitation. You have to know what you're looking for. And you will need a large number of assays if you're looking for different viruses. It's virus specific, generally for high sensitivity. On the chemical induction assays, my lab did develop optimized protocols to be able to induce retroviruses as well as DNA viruses. However, and you can get broad detection, broad activation of these latent viruses, but clearly after they're activated, you may miss unknown viruses because of the limitation of the conventional assays. So I also wanted to highlight that generally we've been talking about testing, but testing is one component of the strategy for adventitious virus risk mitigation. Clearly, you have to also introduce steps to prevent the introduction of the risk, as in terms of using qualified materials, and also trying to know the history, documentation of the history of the cell passage and characterization. And then also, and this is not suitable for all products, but some products can undergo the incorporation of some viral clearance steps to remove and inactivate viruses. And this also results in reduction of residual cellular materials, DNA, RNA, proteins. But these steps may not be amenable to many of the live viral products or products that may be sensitive to these steps, and therefore testing is of paramount importance as a critical safety step. So I wanted to just mention that there are a lot of challenges for testing viruses as adventitious agents. And this is because the viral families, there's a large number of families of viruses. There is high diversity in viral sequences, especially in the RNA viruses. And we recognize there's a continuing increase of the virome, and this is due to the high availability, due to the increase in the availability of high-throughput sequencing, also called NGS, and also with the reducing cost. So the virome is just expanding exponentially. So it's recognized that there is a need for improved analytical methods for broad virus detection. These methods should be able to detect not only the known viruses, but also unknown as well as unexpected viruses, which can be a wide range of viruses depending on the cell substrates, depending on the passage history, the reagents used. So you can get tumor-inducing viruses, latent viruses, unexpected viruses, as well as unknown viruses. And also there's many species, avian and insect. They can constitutively produce a reverse transcriptase activity, and so it's important to be able to evaluate whether this activity is associated with a retrovirus. And so industry and regulators recognized the powerful new technology NGS that has capabilities to detect known and unknown viruses, and this does not need any prior sequence knowledge. And this was actually highlighted with the use of NGS as well as microarrays by a university lab at UCSF, led by Eric Delworth, which was published in 2010, the identification of a porcine circovirus type 1 in a licensed rotavirus vaccine. And then later in 2014, our lab, Helen Ma, used NGS and degenerate PCR assays to discover actually a novel virus, a rhabdovirus in the insect cell line, which is used for baculovirus express vaccines and other biologics in some cases. So I should mention the PCV1, it was an unexpected finding, but it was not an unknown virus. PCV1 is a known virus. It was a known virus even then. It just was not picked up in the 9CFR testing. So highlighting the need for a broader virus detection assay. And I should mention in both cases, routine testing had been done, and additional testing using degenerate PCR assays were done for the SF9 cell line, which was a novel cell substrate at that time. And but these conventional assays and extended assays failed to detect these two viruses that were discovered with NGS. This slide just mentions the various discussions that initiated as early as 2009 related to the use of new technologies for adventitious virus detection. And these discussions actually were related to also the introduction of novel cell substrates and how to address the concerns for the potential of unknown viruses that could be present. And actually, the result of these discussions led to the identification of some of the general challenges for applying NGS for virus detection. Clearly, there was a need for development of appropriate reference viruses and other standards to be used for virus spiking studies in various test materials for manufacturing that could demonstrate the method validation and qualification of NGS and to demonstrate its suitable application of the platform. Additionally, there were challenges at that time for the bioinformatics, which was related to the data analysis, the pipelines in the workflow needed to be optimized, as well as importantly, the need for development of a complete and correctly annotated publicly available reference database. And of course, there were challenges and still existing for the data submission, storage, and transfer related to format and security. And I just want to highlight that it was recognized when you use NGS, it's a nucleic acid-based detection technology. When you detect a signal, you will need a strategy to confirm that it is a true viral hit and then to go on and follow up to determine the biological relevance and significance of the positive signal. You may notice that this is not something that's unique to NGS. This is something you would need to do even if you were using a PCR assay. But there's more chance of picking up these signals when you're using such a broad detection method that can pick up every nucleic acid in your sample. So the much greater need for the strategy for the follow-up of the signal. This just is a very simplistic depiction of the NGS workflow. As you've heard, there are various steps involved in the workflow. They include upstream steps that may have some pre-treatment steps to reduce the background, the nucleic acids, to enhance the signal, to enrichment steps of the particles that you may consider or investigate to enhance the detection of the viruses. And then is followed by nucleic acid extraction methods, and you can have different materials, whole cells, lysates, cell-free supernatant. And again, these would use different protocols, most likely. And then it's followed by library preparation steps, where again, you may have different protocols involved there. And for this step, which is also involves different protocols, RNA depletion may be included, poly-A selection perhaps, cDNA synthesis, of course, and fragmentation for preparation of the library. And then, of course, you have your choice of sequencing platforms based upon the question you're trying to address. You can use short read, Illumina, or long read platforms. And Colette gave very nice details of these. And then, of course, the final is the bioinformatics, and there there are various programs available, analysis tools, and the database is critical for the final results. And also then after this step, it's very important to submit your results in a detailed report for review and evaluation with interpretations. I just want to mention that these challenges were recognized early on by regulators and industry, and we developed a advanced virus detection technologies group. Initially, it was a users group. It became an interest group, and now it's expanded and advanced to a working group, and that's the current status of this group. We have 4 chairs of this group indicated here, representatives from different geographical areas. And the mission of this is to advance the tools for next generation sequencing of viral risk evaluation. And although there are regulators that are participants, myself included, this is not a forum for regulatory questions and answers, but this provides an informal scientific forum for discussions and scientific collaborations, and we focus on NGS, and we have it's meant for knowledge exchange and also for collaborative studies, and this is open to anyone who would like to join to learn about NGS, to bring up general topics for discussion, and as well as to address any challenges by data generation or by discussion. Currently, we have more than 270 international participants, over 60 organizations. They include regulatory agencies, government agencies, industry, service providers, technology developers, and academics. And at this time, we have meetings every other month by teleconference. So I'll try to go through this very quickly, but so as I mentioned, this group, the objectives are to provide an informal scientific forum for discussion, to develop consensus views, to conduct collaborative studies that can generate data that can be used for further advancing the NGS applications and for decision making, to publish best practices, perspective, position papers, as well as to interact with other consortiums. Based upon the challenges that were recognized for addressing NGS applications, there we have different subgroups. Early on, there were individual subgroups A through E for different steps: sample preparation, virus standards, database generation, bioinformatic pipelines, and follow-up strategies. And then as the discussions in the group matured, we then coalesced some of the groups A, B, and D, E, so that currently these are the current groups that are addressing these challenges. This slide just indicates the various collaborative studies that have been completed in green here, and others that are currently ongoing. Some are near completion, some are just starting. You can see the dates here. And this is to evaluate the reference standards and bioinformatic tools for NGS, as well as to compare and optimize experimental protocols. And we're evaluating both short read and long read technologies. And each of these spiking studies have leaders to lead the studies and help conduct and publish the studies. And these are done under a CDA, and once the participants have finalized their commitments to do the studies. So I'm now going to switch to the efforts from my lab towards NGS implementation. We are participating in the studies I highlighted in the previous slide, and these are to evaluate different NGS platforms to determine breadth and sensitivity of virus detection in different matrices. Additionally, my lab took on the, I guess, the task of developing virus reference stocks, since there was a need for it, but no mechanism for sharing funding resources to do this within the working group. So I was fortunate to be provided funds from CBER and my office to develop virus stocks. The first generation of the stocks were 5 reference virus stocks, and they were established through the WHO, but then the next generation are the 7 virus stocks that are currently the WHO International Reference Panel. And the first virus stocks have now been designated as CBER NGS virus reagents. They have both been characterized the same way. It's just, but were manufactured with slight differences in the production. And these are available through NIID, through their BEI repository. Catalog numbers are indicated here for both of them. The CBER virus reagents are provided now currently for establishing and investigating and advancing NGS for implementation, whereas the reference panel of the 7 viruses is being provided for assay for method validation and qualification of NGS. These are available free of charge, and you can get in touch with BEI. I don't know if there's a shipping charge, but there's no cost for the viruses themselves, and they are available for anyone. But you have to register with the BEI, and I can give you more information later if interested. Additionally, we took on the generation of a comprehensive reference virus database. RVDB, currently version 30 is available, and it's available at the University of Delaware, publicly available, free of charge. It's downloadable also, and there's a link and of the proteomic version that's available at the Pasteur Institute. A little bit about the details of the reference of the 7 virus reference panel. These were established as the first WHO International Reference Panel. They are intended to be used as a panel, so you will receive them as a panel. And these were based on a CBER collaborative study with participants of the working group. These have been characterized for infectious titer, particle count, genome copy number, as well as for adventitious viruses by NGS. And this panel, as I mentioned, is specifically provided for qualification and validation studies, and they're available through the BEI resource repository. There's a limit per individual for 1 order per year, and we've recognized that this is sufficient for the studies. If additional is needed, you may submit a justification. And this was generated with 1,000 vials, and they're individually vials so that you can mix and match as needed to create your own genome copy number for detection. And these are the 7 viruses that are listed here based on the different properties. This lists them more, I guess, by in a pictorial manner. They represent different particle size, envelope properties, genome topology, double-stranded, single-stranded, linear, circular. So all different genomes are represented here because we can't represent every single virus that you want to look for. So this represents the different types of genomes because the technology is based on detection of genomes. So these are live viruses, and they're live so that they can test the entire workflow from extraction to the bioinformatics. As I mentioned, there are 5 viruses that are now provided as CBER reference reagents, and these are the 5 here. And then the 7 expanded by adding 2 more viruses, but the 2 are independently manufactured virus stocks. Okay, so as I mentioned, these reference viruses have been extensively characterized and tested. I'm not going to go through the list again. Stability studies have been done. The reference virus genomes have been sequenced and variant analysis performed, and these were by Perushin in my lab. And then we also did the NGS adventitious virus analysis, as well as we've done ddPCR to also quantify the not only the genome copy numbers, but also the host cell residual sequences in the virus stocks. Okay, so now I'm going to switch to our development of the RVDB, and this was developed for addressing the limitations of the public databases that we recognized, and this was early on during our discovery of the novel rhabdovirus. So we started this effort in 2013, and this was done in discussion with the subgroup C, the database subgroup, but but it was prepared in-house by our lab and is being maintained by our lab. And then this included so this indicated that there were deficiencies in the public databases. The NCBI viral RefSeq neighbors had underrepresentation of partial viral sequences, endogenous retroviruses. The NRNT had all viruses, but they were overwhelmed with cellular and uncharacterized sequences. And the protein database only contains complete protein sequences. So, so then we undertook to develop this comprehensive reference virus database to facilitate broad virus detection. The initial database was created by Norman Goodacre in my lab and published in 2018. And so this contains all viral sequences, regardless of size, includes endogenous viral and retroviral elements. It has reduced cellular content. As I mentioned, the latest version is version 30, and this is being currently developed and maintained by Perushin in my lab, and is hosted at the University of Delaware by Sean Paulson's group. And we have the prodict versions at the Institut Pasteur. So this provides high diversity of viral sequences to increase likelihood of novel virus detection with reduced non-specific cellular hits, resulting in less data volume for bioinformatic analysis and less computational time. It is currently being updated twice a year, and there is ongoing work on annotation of the sequences to remove misannotated sequences to enhance virus-specific detection. We recognize that some of the sequences may contain non-viral sequences, and because the sequences that we obtain, they are directly obtained from NCBI. We do not modify them in any way. So what we have currently done is we've undertaken to try to identify those sequences and to have an automated pipeline available at the Delaware's on the RVDB website that will indicate if you get a hit, is it to a non-viral sequence, or in that way, it will help with your follow-up for virus detection. It's in 4 different formats for use based upon the question you would like to address, and also is downloadable. This is the current, as of September this month, the current usage of the global usage of the database. It is growing. And then we also like feedback on the database to understand, if there's any problems with it or any complex issues related to it, or if you identified some sequences that we need to annotate. So I'm now going to focus on the current status of NGS in our office. There's general acceptance for using NGS as a replacement assay to replace the in vivo assays and the PCR assays. And the in vivo assays do include the antibody production assays as well as the in vitro adventitious virus assays. We also accept the use of NGS as a supplementary or a replacement assay for the in vitro cell culture assays, as well as I said, the follow-up strategy is important. So we do expect that you will, if you have hits, you will provide the interpretation of those hits, And some of the points considered there would be to help determine the biological relevance and significance for decision making, would be, can the results be confirmed with another assay? Is there a complete virus genome? Are there virus particles present? Are they infectious? Is there a replication competent virus? Can the nucleic acid or particles be quantified? So the general expectations in OVRR submissions is to use the reference viruses, the WHO reference panel or equivalent, that has been characterized with the same criteria for interpretation of the results. There's expectation to use non-targeted NGS bioinformatic analysis using RVDB or an equivalent for broad virus detection when you're replacing the general virus detection assays. And the method, of course, should be appropriately validated for its intended use. This includes method validation and matrix-specific qualification verification using suitable reference materials, such as the virus standards and a comprehensive virus database, to demonstrate the limit and the breadth of detection, and then to please submit detailed reports and SOPs. I just wanted to point out that we, as well as in the ICH, as well as the Pharmacopoeia, the European Pharmacopoeia, does not expect a head-to-head comparison of NGS with an existing method for replacing the in vivo assays. And this is detailed with the chapter that's coming out in October from the Pharmacopoeia, the 2.6.41. And so you'll have more details there. And so introducing HTS can facilitate increase in the efficiency of the testing time, and it of course reduces animal use for ethical reasons, and it provides superiority readouts for LOD specificity, repeatability, and accuracy. The current cell substrate and viral safety guidance and guidelines already provide flexibility for using alternative approaches with broad virus detection capabilities and fit for purpose. This includes our FDA guidance, although it's from 2010, and we're currently in the process of updating it. It already has the language to provide flexibility for using other assays that fit for purpose, as well as the WHO, even the 2017 European Pharmacopoeia. And of course, the NGS has been now introduced into the Q5A R2 document in 2023. And I just wanted to briefly highlight some points in that document. The document has been updated to reflect the current scientific knowledge and biotechnology advances, and specifically related to introducing new assays and alternative analytical methods such as NGS. It encourages the use of NGS to align with the 3Rs initiative to reduce animal testing. And I've already mentioned, and we're aligned with the Q5A also in replacing NGS for the in vivo assays. And it also indicates that a head-to-head is not needed for in vivo and in vitro. And it's recognized that these nucleic acid-based technologies have limitations. They cannot distinguish between infectious and non-infectious particles, and therefore detection of a signal may need a confirmatory result, a test with an infectivity assay for risk assessment. So this just indicates the potential applications of NGS for testing to mitigate risk of adventitious virus introduction, to monitor the absence of extraneous viruses during production, for detection and characterization of extraneous viral sequences in the final product, and also for characterization of viral vector sequences. I would just sort of come near end of my talk to indicate that the work hasn't been finished. There's still more work for routine implementation. We have continued in-house efforts, and the working group efforts are continued for evaluating performance and NGS in different matrices. Currently, my lab is involved in a collaborative project for optimization of pretreatment conditions to increase sensitivity of virus detection in complex matrices that is supported by the Gates Foundation through NIMBLE. And we also are focusing on developing SOPs and reference data sets for broad NGS implementation. And this data sets is referred to for having some reference material for the bioinformatics pipeline, actually. And then there are opportunities for developing other standard materials, even for the wet lab, VLPs, virus-infected cell line synthetics, as well as need for data enhancements for increasing accuracy and for virus detection and reducing follow-up of false positive signals. I think I also want to mention that I should have included that we also are developing. Sandro Fuentes in my lab is working on developing cell line standards that can be used for spiking studies to evaluate cell-based products as well as cell substrates. And her effort is supported by Andrea Kennard in my lab they're using single cell sequencing also to characterize the virus infection in the cell line standards. And with that, I have a list of references that are here, and that ends my talk. Thank you very much.
Thank you so much, Arifa. We have about 10 minutes for questions from the audience. If you have any questions, please do add them to the chat now. There has been one that's already been submitted, one that will be addressed tomorrow during our panel discussion, which Arifa will be joining as well. But for the first question, are these reference viruses intended for use in regular NGS analysis as positive controls for assay validity?
Yes. Well, yes, they are intended. The 7 virus panel is intended to be used to validate the NGS method and qualify, and to use for product-specific qualification of the method. And they are publicly available. If you're doing some investigational work, then we will direct you to use the CBER virus NGS reagents, just because I mean, they are limited. So we don't want to deplete the stock. The 7 viruses were developed to replenish the stock of the 5 viruses. But again, so we've sort of still provide the 5 viruses for establishing NGS, and then for then the 7 are there for actually doing the actual method validation. I hope that answered it.
Great, thank you. Is there any harmonizing SOP to ensure standard data coming out of the different labs?
So that is the goal. We would like to be able to provide such SOPs. So that is sort of, you have all that list of different collaborative studies that are ongoing, and each of them has a large number of participating organizations with different workflows. And so once the data is generated, the goal is to try to see if a common SOP can be developed, or if not, which is a big ask, then at least we can highlight the critical steps or critical points that need to be considered by the user for optimizing the NGS protocols. But yes, that would be great if there could be an SOP, and but there are we are working towards that, and these type of webinars are we also try to have some training webinars, but definitely I mean, more work needs to be done, and a lot of it is based on generation of data towards developing some SOPs that hopefully will be based on the optimized conditions.
Great, thank you. For adventitious agent testing, what depth of sequencing read is acceptable?
Okay, that's a very good question, and that's a question that has been there for the last, I guess, 10 years when we've started working on NGS. How much is enough? And we're also, I mean, I can throw out a number. I mean, clearly the more depth, the better. But at what point do you not, you at some point you start seeing more background than your viral sequences. And so, and there may be different considerations for this. I mean, initially, if everybody was using one platform, you can say, okay, you want to get, at least 3 or 500, thousand reads. But but then the platforms are now different. They give different size of the read. So and you can get with the nanopore, you even have less reads, but you have longer reads. So I guess the short answer would be you use the reference viruses to demonstrate the sensitivity of detection based upon the platform you're using. So even if you get less reads, if you have other steps in your protocol that enhances the virus detection, that may be sufficient than someone who's getting more reads without those um, um, pre-treatment steps or enhancement steps for virus detection. So that's the reason it's important to demonstrate the sensitivity and breadth of detection using the reference viruses, so that's sort of a, I guess, comparator.
Great. Thank you. And as a follow-on to that question, what about coverage? How long should the read be to be considered a positive?
So it's not, I guess maybe you're asking how many reads would make a positive result, because I mean, you can ask for the Illumina, the reads may be very short. For another tech platform, the reads may be longer. So, for Illumina, if you have a short read, is one read sufficient? Or how many reads do you need to say the result is positive? So this again is something that we're hoping our studies would help with. So this is in some, I guess I hesitate to say one read, cannot be a positive, because we found one read can be a positive, but it could be, it depends on also the follow-up result. Generally, when we were saying one read is a positive, we were still using the 454, so the read length was a little bit longer with Illumina. Or if you're using the a very high throughput like the NovaSeq, if you've got millions and millions of reads, in the context of that, would one read still be a positive? So I think the problem is you get a signal and it's hard to detect it. It's hard to ignore it. So and I know different labs, based upon they have different labs have different criteria based upon their background signals and based upon their interpretation of a real positive hit as to what their cutoff criteria might be for a positive result. So I think that's where each lab, and again, I'm sorry, there's no established criteria at this time. Okay, that this is the cutoff for your result. I mean, clearly you want to, and you'll hear more about the controls, you want to always have negative controls in your sample so that you can, so if you get a few reads, then you can see if my negative control is absolutely there's no background, then then you do need to consider why if those reads are real viral reads, or are they something that is being picked up because it has some cross reactivity to some other non-viral sequences. It could be, is it a low complexity sequence? So I think those considerations have to be done, but we're also again hoping once we have these collaborative studies completed that we will be able to further answer, have more, I guess, defined answers for these questions. But at
Thank
this
you,
time,
Arif.
we are accepting, we're reviewing the NGS data in our submissions, each case by case, and it's based upon the totality of the results that we receive and the interpretation that you give us, and then we evaluate the interpretation to see whether, it's valid or not, or we may suggest you may need to do other additional analysis if you haven't done it. But in our office, we are not asking for the raw data. I should clarify that. We are not, and each office may be different, but we are not asking for your raw data because we're going to repeat your results. We ask that you provide us your, maybe in an Excel sheet, the hits, maybe if you can nicely provide the visual coverage and stuff, that's also very nice. But we review the data that you submit just like you would review it in-house. So we have those capabilities. And then if there are additional questions or suggestions, then we will go back to you and ask you to repeat something. If there's a particular question, then we could ask for a specific sequence just to try to help you resolve something. But we don't normally repeat the data the results that you're submitting. We review them, we interpret them, and then we provide feedback on them.
Great. Thank you so much, Arifa. And with that, Arifa will be part of our panel discussion tomorrow. So if you have additional questions, there will be an opportunity at that point to ask those at that time. Now I'm going to move us on to our next two presenters. So this is from Millipore Sigma. Bradley Hasson is the NGS Director of Operations, and Leila Diaz is a technical consultant, and they are going to walk us through a validation case study for NGS. So Brad and Leila, we will hand it off to you now.
All right, thanks. I think we were just sharing our screen there, so yeah.
Okay, can everybody see me, hear me, and see the screen with the slides?
Yes.
Great.
Yes.
Thanks you very much for Path for inviting us to speak, and thanks to Colette and Arifa for setting the scene for our presentation. So what Brad and I will go through for the next 30 or so minutes is a more practical approach to the concepts that were presented in the first two presentations. So we're going to talk about the validation of a next generation sequencing method for virus detection, and we will include some data from a case study based on that method. And so if we look at the makeup of the presentation, again, I will go through the virus detection method description, validation approach, building on the concepts that again, Colette and Arifa presented earlier. And Brad will take over and discuss the results from the case study more specifically. So I want to start off with a little bit of regulatory guidance. I know it was touched upon in the previous presentation, discussing ICH Q5A, the virus safety chapter, that sort of gave us permission to use NGS to replace assays like the in vivo assay and the in vitro assay for virus detection in certain situations, especially for the replacement of animal-based assays. But what we were missing, and what this regulatory guidance that I presented in front of you gave us, was a more detailed approach to what those assays look like and what they should contain, as far as specific criteria that regulators may be looking for, and as well as a validation work-through, essentially, of how to make these assays GMP compliant from a validation perspective. And so this the European Pharmacopoeia Chapter 2641, high throughput sequencing for the detection of viral extraneous agents, is currently in draft. The final will be published in October 2025, and it will become effective in April 2026. And again, it provides guidance on the method validation, including the critical parameters to be evaluated, as well as the selection of spike material to demonstrate limits of detection, controls that should be included, and things like that. And so that really has made it so that we can all use a sort of a standardized method to detect viruses. So it's going towards that goal of making these a little bit more standardized, so that when regulatory agencies see different methods from different places, there will be some sort of commonality amongst all of them. All right, so the NGS method for virus detection. In this case, as Colette actually introduced the concept earlier of transcriptomic versus viromic, in this case, we're going to be discussing the viromic assessment. So a method designed specifically for that. So in this case, it sequences all nucleic acids in this test sample to detect both RNA and DNA viruses. In the case of this particular method, there's a nuclease sample treatment to increase the limit of detection. So that's something, it's a pre-treatment that you can perform on the test material itself. That again helps in that virus detection method, in the virus detection realm. It's suitable for complex matrices such as virus seeds and virus harvests. So we're talking about the bulk harvest from a vaccine production or your seed virus as you're characterizing that initial raw material. It's an Illumina-based platform, so I'll be discussing that specific platform. So it's a short read sequencing. And just keep in mind that these assays are designed for broad detection coverage. And we'll go a little bit more into what this assay is intended to replace in this specific case study. But just keep in mind that the in vitro assay and the in vivo assays, especially the in vivo assay for inapparent viruses, are broad detection coverage viruses. And so you want to cast a wide net as to what viruses you're looking for, so you're not biasing the detection of any specific viruses with a targeted approach. So just wanted to set the scene for that. So, I have the individual steps of the NGS workflow at the top. We've seen two different versions of that already, but just to go through a little bit on the specific considerations that you need to have for each one of these steps. Again, you have your test material. In this case, it's going to be a virus bulk harvest, which will contain process-related impurities in it that can be that can interfere with the ability to detect viruses. So you want to see if you can do any pre-treatments. Like I mentioned, we use a nuclease pre-treatment to digest any DNA that's not encapsulated in a virus, or any DNA or RNA that's not encapsulated, to again reduce that background noise from the initial input. You can also centrifuge, do centrifugation, and do some other purifications to your sample to reduce that background. Next, you move on to DNA and RNA extraction. So this is what needs to be tailored specifically for your method. So in the viromic assessment, we have to extract both DNA and RNA. You can do them together or separately. It's up to your specific workflow, but just make sure that that extraction step is geared specifically for the type of assessment that you're performing. And that, of course, leads into the DNA library preparation, which again has to fit the purpose of the assessment that you're doing, as well as the test material input. It's usually dictated by the platform that you're using. So in our case, we're using for this case study, we use the Illumina-based platform DNA library preparation kits. But again, it's dictated by that platform, so that you have a good quality DNA library that goes again into the sequencing. And that concept was brought up earlier in the presentations as well, that you need to have good quality input in order to get good quality sequencing out. And so once you go into the sequencing, you need to make sure that you have, again, a good quality output out of that into the data analysis. And the data analysis is one aspect that, again, is very specific to the method that you're using. In this case, obviously, we're detecting viruses. So you want to apply, you want to make sure that your bioinformatics pipeline can extract good quality data from your, sorry. Oops.
Sorry.
Oh, I'm sorry, we're having a little bit of technical difficulty here. Okay, so you want to make sure that your bioinformatics analysis can extract good quality data from the sequencing, so that then it matches it to, or it aligns it to, a viral database. And the viral databases were discussed earlier. You can use the RBDV database that Arifa spoke about. NCBI databases are also available, or you can curate your own, so that you have your own database that you can maintain. And that's an important component to point out as well, that you want to maintain these databases so that they are updated regularly. New sequences are deposited into the public databases regularly, daily, almost daily. And so you want to make sure that you capture all of those to increase that breadth of detection, because remember your ability to detect a specific virus is only limited by whether it's in the database or not, or whether related viruses are in the database or not. So any novel or emerging viruses need to be captured as well, and that is done through the update of that database. You also want to have, in your data analysis, you want to have a counter screen. So now that you've aligned your references, your virus sequences, and you've obtained those virus sequences, you want to make sure that they are truly virus sequences, that they don't have homology or high level of homology to a non-virus sequence. And so that's what that counter screen is for. And you can use non-viral or viral databases for that purpose as well. And so now, after the counter screen, you have a list of what are your true potential positives, and those need to be put in context. And that's where the scientific analysis of your data, of your output, comes in. You want to make sure that you look through all of those potential positive signals and put them in context of the system that you're working in. Is it related to the virus input? Do you need to subtract? That's one thing I neglected to mention earlier. Do you need to subtract any sequences from consideration because it's part of your product? You have a virus product, you want to subtract those sequences. But then you also want to make sure that you review the output to make sure that it's not related to anything that could be in the system, from the, anything from the cells, anything from the product itself. And that is where that expert scientific analysis comes in of the data. And so you want to have those three components: the initial alignment to the database, the counter screen, and the scientific review, to come up with with a result, a report for your final data. Sorry. All right, so now we're going to talk about a little bit about the validation approach. So as actually somebody mentioned earlier, you need to demonstrate that this method is suitable for its intended purpose. You need to consider the sample type and the testing approach. And again, those two components, we keep repeating that same theme of the sample time and the testing approach, because that's what makes each individual, each one of these assays unique, as from a NGS workflow perspective. And so, you want to make sure that all those that it is suitable for that. Virus detection methods are qualitative limit tests where specificity and sensitivity of the method need to be assessed, and I'll discuss those in the next few slides. So you need to have an end-to-end validation of all the steps in the process. But as I think Colette mentioned earlier, you can also subdivide each component of the process into modules to establish criteria to demonstrate the performance consistency of each step before moving on to the next one. So they serve as sort of gated criteria, so that that you have a minimum quantity and quality for nucleic acid yields, before you move on to the DNA library preparation, because you've demonstrated that that amount of RNA or DNA will yield an appropriate amount of, acceptable, that would yield an acceptable yield of library size and fragment range and amount. And so once you have, again, an acceptable library criteria, you move on to the sequencing. And there you can look at things like quality score for the sequencing data. Depending on the instrument that you're using, you may want to look at cluster density if that's applicable. Again, because you want to make sure that the output of that sequencing is of high quality enough to move on to the data analysis. And there you want to make sure that your bioinformatics pipeline can run can run the analysis on a regular basis with no errors, and you want to establish what those parameters are for demonstrating that it runs with no errors. And the advantage of this knowledge validation strategy is not only to make sure that that you, in this lengthy multi-step process, that you're not that you are getting quality data out of, quality yields out of every single step, but also it gives you the flexibility to update with the moving technology. You heard earlier today about, all the different versions of NGS and how things are improving, and we expect things to move forward with newer technology. And something like this allows us, a modular validation allows us to change a specific step in the process without necessarily having to revalidate the entire process. Illumina updates their DNA library preparation kit. We can just update a new kit, update the validation for the new kit, and then verify that it still works within the validated end-to-end process. But we don't need to revalidate the extraction or revalidate the sequencing as well. So it does provide quite a bit of flexibility in that respect. So now I want to talk about specificity and sensitivity, because these are essentially the parameters laid out in the ICHQ2 guidelines for validation of assays. And this is a qualitative limits test where you want to establish that you are that it's specific for what you're looking for, and that and you want to determine its sensitivity. And so from an assay specificity perspective, you can look at the recovery of positive controls. And I didn't mention those earlier, and I should have, where you can, in the workflow, you can apply different versions of positive controls. You can apply nucleic acid, reference nucleic acids. You can apply non-related viruses that aren't likely to interfere with your ability to detect the viruses that you're looking for. It all depends on the purpose of your assay. But again, the recovery of those positive controls serves as a measure of specificity. If you're doing a virus spike, which I'll talk about in the next few slides, you want to make sure that you're able to recover those virus spikes in the assay, and that the assay is able to detect those when you know that you've included them in the sample. And then, of course, no recovery of virus spikes from the unspiked samples. And so all of those form part of that specificity assessment. You also have assay sensitivity. So this is and I saw a couple of questions in the chat earlier about the limit of detection. So this is where, especially for an assay like the viromic assay, where we're looking at a non-cell-based sample that has been, produced in a cell system, and it has media components, it has cell components, it may be it has a virus product in it, and it has the residuals from the host cell all mixed into that test matrix. That has the ability to influence the level of detection of viruses on a sample specific basis. And so that needs to be assessed individually through a spiking study. And it is performed for an end-to-end analysis. So you perform it throughout, using the entire process to make sure that you're able to not only extract those spike viruses, but also detect them using your bioinformatics pipeline. And so it goes through the entire process. And how that is So I want to take a step back, and I know Arifa introduced the concept of the spiking materials. So for the spiking studies, in order to demonstrate that limit of detection, the European Pharmacopoeia chapter also recommends the use of the model viruses that were presented earlier, and those spike viruses represent viral diversities, as was discussed, different genomic sizes, different genomes, envelope, not envelope. Sizing is important as well, and as well as chemical resistance. And so all those components give you sort of like a full breadth of detection for your assay, right? So they demonstrate that you can detect different types of viruses that could possibly be encountered. Again, going from extraction all the way to bioinformatics detection of those spike viruses. I should also mention that additional viruses can be added based on a risk assessment. So if your specific product has a component during its production or in the cell line that it was created in that could potentially add an additional risk to the system, you can actually use that additional virus as well, or those additional viruses to set your limit of detection. And then you'll see an example of that during the case study discussion. All right, so how do you determine, how do you incorporate that limit of detection determination into your validation? It's essentially a relatively simple process, as I've alluded to earlier. So you have your 7 reference viruses. You spike them at different levels. Starting, and that truly depends on your specific system and what you know about it. But typically, there's between 4 and 5 levels of detection. You want to dilute the virus spikes to the point where you no longer detect them in order to set that limit of detection in that specific test matrix and put it through the entire process. We often get the question of, do I need to do a spiking study? Is there any data that I can leverage to look at previous products that have been performed, that have been produced similarly in a similar background? And so I wanted to address that concept by showing you this graph at the bottom of the slide here. So this was some spiking studies performed for 2 separate products that were both produced in Vero cells. And you can see the level of viral genome copies for the spike viruses on the y-axis. And also, you can see the different virus spikes that were done. This was done with the 5-panel viruses before the coronavirus and MMV was introduced into the 7-panel version of the virus. And so if you look at the 2 different products, you can see widely different limits of detection. Product 2 is a relatively large DNA virus. Product 1 is a smaller RNA virus. And so again, the virus, the product itself, the production of those products made a difference, even though they were both produced in Vero cells. So they have the same cellular background, but distinctly different limits of detection based on different elements within the matrix. So that sort of illustrates why it's important to establish a limit of detection for your specific product. And so this forms the entirety of the validation. You have your the validation of the process itself, where you want to make sure that you have appropriate input of sequencing. You want to make sure that you appropriately analyze the data and that is all demonstrated in a validated form. And then you introduce the specificity and sensitivity concepts using the spiked viruses. Okay. And now I'm going to turn it over to Brad so he can go through a little bit of data on the case study.
All right, thanks, Leila, and I appreciate all the other presenters as well today presenting. So we've heard a lot this morning. Well, it's morning for us in the United States, but afternoon and evening for others. We've heard a lot so far about the NGS methodology, how to implement it, regulatory landscape. But what I'm going to run through today is a case study, essentially validation of an NGS virus detection method in a live vaccine matrix. And I do want to point out our paper. We did publish this data, and so admittedly, a couple of figures that I'm showing are from that particular publication. But we did do this in collaboration with AstraZeneca, who is using this assay for an in vivo based replacement, and that's on the paper. And I encourage you to read it, read up on it if you're interested. And so with that, I'm going to sort of present this case study, walk you through how we did the spiking study and how we actually implemented next generation sequencing for this particular product. And so for this particular case study, we are doing an in vivo replacement. The goal of it is replacement of an in vivo animal model testing for adventitious viruses with next generation sequencing. The test sample is a bulk harvest material for a quadrivalent influenza vaccine. For the matrix, it is allantoic fluid from 4 separate influenza strains. Those strains are listed there. And then we want to keep in mind that influenza vaccine products, particularly this one, is manufactured using SPF eggs. And so because of that, there are regulations that we need to follow for that. And essentially, the in vivo assay needs to be performed on every single lot because of the origin of the material. And so for this study, we were looking at using NGS. We are using short read technology. So pretty much what Layla was presenting before, a lot of what Colette went over to replace the in vivo assay. But before we go any further with that, what is the in vivo assay? We've all mentioned it, we've all said it, but I don't think that anybody has presented this particular figure to describe it. So I'm glad I'm not being repetitive. We have the traditional in vivo method. It is a well-established method that utilizes animals to detect adventitious viruses within a biologic material. With that, you will essentially follow the scheme in the middle. It uses adult mice, suckling mice, and embryonated eggs. It is subject to high invalid test rates, and that was one of the motivations for moving to next generation sequencing. Now, in terms of the NGS alternative, this has already been described today. It is a broad spectrum virus detection method. It does it has the ability to demonstrate comparability or better virus detection than the in vivo-based method, reliable methodology with objective results, and it is in sync with the 3R methods around the world to replace the use of animal model testing. And I will also mention that we've actually launched our first NGS assay back in 2015, 2016 timeline. There wasn't a lot of uptake of the method then. It was always for the in vivo, in vitro-based replacement, but a lot of the initiatives around the world, like the AVTIC consortium, the studies that were done there, have the incorporation of NGS into the guidelines have helped progress NGS. And as a service provider, we do generally see an uptick around the world in next generation sequencing adoption of those particular methods. So with that, I'm going to go into our study design where each individual vaccine component was tested separately. So this essentially gave us the 4 separate samples. The viruses that we used for the spiking were actually the original panel. This study was done about 2 or 3 years ago to assess the limits of detection, like Layla was going over before. And so we did use the EBV, RSV, FELV, PCV, and Rio. We also added an additional virus, was BVDV, not because BVDV was a particular concern, but because that particular family of viruses was a concern for the client. And so we did incorporate this. And so we essentially moved forward with 6 viruses for the spiking study. The concentrations that we used were in line with those that are generally tested in the industry. We used a 1E6 viral genome copies per mil spike, a 1E4, a 1E3, a 1E2, and an unspiked sample. We also perform replicates at each level to ensure consistency of performance. So assessing this within the entire matrix. Now, in terms of the method, we do use our fully validated method. So this is a method that was validated with the modular approach. And so like Layla was discussing before, the spiking study is essentially looking to complete that validation within the context of the client's product. We did perform it to GMP compliance to fulfill the regulatory expectations. We did use the WHO standards at the time, which were the original 5 viruses plus the BVDV. And again, we did use the NextSeq 2000 system. Now, in terms of the results for this particular spiking study, it was a very large study. We had 32 data sets. The reads per data set range between 190 million to 640 million. Now, while that may seem like a large range, it really depends on the sequencing metrics and batchability on the sequencer. But what we do is we normalize that data to hits per million. And so that way your sequencing metrics don't really have an impact in your detectability. Our average read size was in line, as was all of our runs. All of our runs were validated, and the cluster density was in line with that of a pattern flow cell. Now, when we come to criteria for detectability, we do say that one hit is detection of a particular virus, and we demonstrated that, and I'm going to show the data in a second. And then we did require that 7 out of 8 replicates, so we did test the 4 viruses and replicates at each one. We did require that 7 out of 8 were required to produce a positive result to be called the limit of detection for this particular study. Now, when we come to the methods, I'm going to go through these each sort of individually. With our spike detectability at the 1E6 viral genome copies per mil, this was essentially our positive control. We actually did a fair bit of work prior to getting to this study, where we knew that the 1E6 was going to show detectability, and so this served as our positive control with all that passed. We didn't perform replicates here because this was the expectation and the criteria moving forward. But then we've got the 1E4, 1E3, and 1E2. And you can see from the data that for the 1E4 viral genome copies per mil spikes, all spikes were detected in 7 out of 8 replicates. You can actually see with Rio, with the 3rd sample, we started to lose it. And I'm going to discuss that in the next slide about some of these viruses that may be a little bit more difficult to detect. In the 1E3 viral genome copies per mil, we did lose PCV and Rio, where we did not detect 7 out of 8 replicates, but we did detect in EBV, RSV, FELV, and BVDV. And so this was essentially the limit of detection for those viruses, but PCV and Rio were a higher limit of detection. Now, of course, we're using this for specificity as well. Oh, sorry, before I say that, at the 1E2 viral genome copies per mil, we did not detect any spikes in 7 out of 8 replicates. Now, the slide was getting a little bit busy, so I didn't put the unspiked samples, but for the unspiked samples, we did not detect any viruses. And that serves as our basis for specificity and our basis for determining that one read is considered to be a positive, because we did not see any in the unspiked samples, and we do see them in the spiked samples. Now, when you combine all of this together, what do we get? We essentially get our final LOD, which is in the yellow box, where we have final detectability of EBV, RSV, FELV, and BVDV at 1,000 genome copies per mil. And we have our final result showing detectability of PCV and Rio at 10,000 genome copies per mil. Now, if we think about some of these viruses that were spiking, like Arifa was mentioning and Layla was mentioning as well, they do represent all of the different physiochemical properties of all known viruses. So you have double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA, large viruses, small viruses. It's pretty much all-encompassing. And so when you look at these viruses, PCV is a very small single-stranded DNA virus, only 1.8 kb. And Rio virus is double-stranded RNA segmented virus, also small as well. And so these were the 2 most difficult to detect by using molecular-based methods. We see that in PCR-based assays as well. Whereas the other viruses, EBV is a nice hardy double-stranded DNA virus, RSV, FELV, RNA-based viruses, BVDV as well. And so we see better detectability of those viruses because of the different virus families and different detectability that we do see. And this is pretty much seen across most of our studies. It's been presented in industry forums as well, where we generally see lower limits of detection for these particular viruses, although we are, as an industry, continuing to optimize the methods to look at what we can do to boost detectability of these particular methods. And a couple of conclusions from our spiking study here is that NGS was able to detect representative virus spikes with a suitable limit of detection in line with previous matrices published through some of the worldwide studies. Generally, we're seeing limits of detection between 1E2 viral genome copies and 1E4 viral genome copies, again, depending on the matrix, because like Layla was discussing before, the matrix has a large impact on your detection of the viruses. If you think about other methods like PCR, where you're amplifying small regions, you're essentially boosting that above a background via exponential amplification, whereas NGS-based methods don't have that. And so the background actually does have a very high impact on what your limits of detection are. Again, we have the higher detectability in PCV and Rio, not unexpected given the nature of those two particular viruses. And then for this, we look at how many reads do we need to achieve in order to maintain that limit of detection? And we set that number to run the assays. Now, in this particular study, it's approximately 100 million reads. I think it was actually 130 for this particular study that we needed to maintain in order for that limit of detection to hold true. So if we drop below that limit of detection, because we have that hit per million assessment, we don't accept that particular data for the sample and need to add sequencing runs or number of reads or however we address that in order to reach that minimum number of reads that we confirmed during the validation. This also speaks to the question that came up before, how many reads do you need? Well, if you're using these viruses in a spiking study, you can set that number based on the detectability and the limit of detection that you set. Now, that spiking study it's great. It's all well and done. We've published on it certainly with AstraZeneca, and we're moving that forward. But when you think about implementation within the context of a regulatory filing, really your spiking study is the first step. It's probably only about 20% of what you need to do in order to implement NGS. So there are other steps, of course. So first, the first step here is the validation and the sample matrix qualification. This is the spiking study. This is your assessment of the sample, ensuring that all of your controls perform as they expected, with no hits in your unspiked samples, hits in your spiked samples, and setting that limit of detection. Then comes your regulatory submission approach. And we always encourage early engagement with the regulators, especially using NGS. We've seen over the years, and we've been doing this for a while, that as NGS has gained in popularity, we see more consistency in questions coming out of the regulators. And we sort of have a recipe for success. And that's my plug for CROs right there, is we've done this quite often. And so, no matter what CRO you go with, we've all, worked with clients on regulatory submissions within the context of NGS, and we've all addressed health authority questions. So with your regulatory submission approach, you do have the validation package, as well as your product-specific qualification report. So that product-specific qualification report is owned by the client, but the validations are generally owned by the CROs. But if we're talking about the FDA, I'll give that as an example. We do have our biologics master file with the FDA, and all of our methodologies, including our reports, our SOPs, our bioinformatic algorithms, everything is stored in that biologic master file. So we simply provide the client with a letter of authorization for the reviewer to access that. And so you would provide, that letter of authorization to access the generic validation, and then the second layer, which is essentially your qualification report. Now, for other jurisdictions around the world, we actually work directly with those agencies to provide anything that they're asking for. So we're fully supportive, as are all CROs, of providing that information to the regulatory agencies. And so we work directly with them to get them everything that we need. Other things that you'll need to do, essentially a contamination control assessment, any relevant risk assessments that need to be introduced for introducing NGS. Is it a substitution within the context of the AstraZeneca paper? We were substituting the in vivo assay for a commercial product. Now, is it a new product? Then you'll need to conduct the appropriate risk assessments and make sure that NGS is, essentially doing the same thing as the traditional methods do, and you have a complete comprehensive safety package that you're submitting to the regulatory agencies, and they evaluate the use of NGS within the context of that full package. Next come the questions. And there are questions for NGS. I'd be lying if I said you're going to submit NGS and it's just going to be a blanket acceptance. That there are questions, but the questions are becoming more consistent now, both within the different agencies and within the same agency. And we're able to essentially provide the information that's being asked. And then come approvals in specific regulatory jurisdictions, and then implementation. And if you think about approvals, if you're filing in multiple jurisdictions, and we do have experience with this as well, as you're going to get your approvals at different times. So how are you going to phase in NGS over the life of your product if you're waiting potentially 6 months or a year between approvals in different regulatory jurisdictions? And these are all things that you need to consider when you're implementing NGS. So there's a lot involved after you do your spiking study. Again, we're fully supportive as our all CROs if you decide to go down that particular path. But again, we do encourage engagement with the regulators early on in the process of using next generation sequencing. Now, a couple of takeaways just in general here from the talk that the case study that I gave and the talk that Leila gave previously. And NGS is well-supported alternative to traditional testing for adventitious viruses. With that, number two, the guidance documents do provide information on the method and the validation. And so we do see full support within ICH Q5A for using next generation sequencing as a replacement for in vivo, and it opens the door for using it for other methodologies as well, like Colette was mentioning, using it for PCR-based testing, using it for in vitro-based testing, and so the incorporation of that is really important. And then the guidelines that are coming out, EP 2641, which will be finalized in October and go into force in April, provides consistency in requirements for validations. And then number three is NGS has been successfully implemented in vaccine products. Oftentimes people will ask, well, have you implemented it within the context of regulatory filings? The answer to that is yes. We certainly have, as I demonstrated in the case study, but there's many more clients that we've been working with over the past 10 years or so that have used NGS within the context of regulatory filings. So it is being accepted, in our experience, and we're moving forward with that. So that's my last slide, and with that, I appreciate everybody's attention, and I guess Leila and I can take some questions here. So I'm going to stop sharing so we can see.
Great. Thank you, Brad and Leila. Very good overview of the validation package. We did have several questions that came in during your presentation. After this question and answer period, there will be a more general one for any residual questions for any of the other presenters. So if anyone in the audience has specific questions for Arifa or Colette, you feel free to put them in the chat box as well, and we can direct them to those individuals. So for Brad and Leila, the first question, at analytical development phase, what comes first, setting the library prep or the bioinformatics pipeline?
So yeah, good question. I mean, these are all done in a modular format. I think Colette alluded to this, as did Leila, where you're looking at extraction, and you're gating that. You're ensuring you're validating that method to ensure that you have suitable material to then move to the next part of the process. Then you have library preparation. Again, you're ensuring suitability of that library preparation prior to moving to the next part of the process. You then have sequencing, and then the bioinformatics, where you're designing that algorithm as a module. And so each of these are done separately. So you can essentially, work on both of them. in parallel at the same time. Now, once you've validated your modules, you do need to demonstrate that they work together through an essential qualification of those methods. So you would perform the end-to-end analysis at that time to make sure that everything is sort of working together. One of the reasons why NGS is really validated in a modular format is to allow for changes in the rapidly changing technology. So for example, over the years, we've seen different sequencing kits. We see different sequencers come out. Now, do you need to revalidate extraction, library preparation, and bioinformatics because a sequencer came out? No, but you can validate that sequencer and then confirm end-to-end performance of the method. So I just wanted to point that out as well, that you can, definitely work on them together.
Perfect. Thank you. How many replicates do you recommend for QC release testing?
So during the validation and the spiking study, actually, EP2641 is going to provide a little bit more clarity on this. Now, for the spiking study, case study that I showed, we did 2 replicates, but we did 4 different samples. But moving to EP2641, it's asking for 3 replicates, and it's asking for positivity in 3 out of 3 replicates. That's going to be in the new guidelines for those of you that have seen the draft or will see it afterwards. And so during the spiking study and during the validation, we test 3 replicates to ensure that we're getting 3 out of 3. Now, when we move to the test sample, we actually only test 1 replicate of that because we're justified in doing that, in that we've demonstrated consistency of testing 3 out of 3 replicates in the validation. And because we require those 3 out of 3 to be positive within the validation as well as the spiking study, we can now move to testing 1 replicate of the test sample in the routine testing. Of course, as long as it's all supported by the matrix qualification.
Great. Thank you. There's a statement on here that NGS detects both fragmented infectious and non-infectious genetic material, whereas the in vivo methods detect only the infectious genetic material. Any comment on that statement?
Yeah, it's true. So if you detect something by NGS, it doesn't tell you if it's infectious or not. Now, if you look at the in vitro and in vivo based methods, it doesn't tell you what it is, right? So you sort of almost have a complement there, where if you detect something, those assays would tell you if it's infectious or not. NGS would tell you what it is. Now, if you do detect a signal by NGS, I believe multiple speakers had mentioned this. If you find an actionable signal, regardless of what your criteria is, that needs to be followed up. Now, depending on that signal could guide what your follow-up is. It could be additional molecular-based methods, but it could actually be an infectivity-based study to see, what it is. And you may actually have already done that if you have performed the in vitro study with your thing. But yeah, that's right. And NGS isn't going to tell you, especially with short read technology. Now, if we start getting into long read technology, you can get more indications. If you have millions and millions of reads, we always talk about that worst case scenario where you're detecting 1 read or 2 reads or a handful of reads. You're not going to be able to tell if something's infectious based off that. But if you see something with millions and millions of reads, and I mean, we've certainly had those case studies come through us, then you can infer a little bit more information, and most likely NGS won't be the only assay that you saw that with.
Great, thank you. So there's an individual that's worked on the ion torrent and the nanopore sequencing systems for targeted infectious disease diagnostic panel development purposes. In their tests, they considered at least 10 assembled reads of at least 100 base pairs on those systems to be considered as a positive for the adventitious agents that were studied. You had mentioned that 1 hit would be considered a positive in this validation study. How long does that 1 assembled read need to be considered a positive?
So I'll go back here to some of the spiking studies that we have done. Now, when we spike these at a very low level, sometimes you'll only see a handful of reads. Sometimes you'll see one read, sometimes you'll see 10 reads. And so we don't put any criteria around that. Our criteria essentially for a positive read is one hit, as long as that's supported by the validation. So one hit. Now that read has to have certain specificity, so we do require it to be specific to a particular virus or virus family, and have a certain level of homology over approximately at least 100 base pairs. Because once you hit that 100 base pair length, you've definitely got enough specificity. I mean, you can design primers that are specific to certain viruses that are only 20 base pairs in length. And so when you're searching for adventitious viruses, we do consider one read to be a positive.
Great, thank you. In a quality control regulated environment, assay validity is the foremost requirement. What are the validity criteria that are taken into consideration during interpretation of the assay outcomes?
Yeah, no, another great question here. And so we do have those gates, right? So we have each one of the modules. And so we do monitor as we move through. So after each one of those modules, there is a quantity and quality assessment. And I believe Colette was talking about that as well, that you're required to meet in order to move to the next step. So we are assessing the behavior of the sample in the test system throughout the entire process. Now, our validity criteria does require quality reads off the sequencer. It may be just the quality, it may be cluster density as well. Again, it all depends on what system you're using. Like what is a valid metric to assess the performance within the particular system that you're looking at. And then once we get there, we have our bioinformatics algorithm. Now, depending on your assay, you may have controls. One of the controls that is in EP 2641, the new chapter, is an end-to-end control. So you spike something in prior to extraction, you look for detectability on the back end. So that really monitors the entire process. So it again, validity criteria is going to depend on your particular validation, your particular additional qualification as well. So in the case where you're looking to determine number of reads, you know, in our case, we had to hit a specific number of reads in order for that LOD to hold true. If we were down at 50 million reads, then that LOD would not hold true. So that could be another criteria. So again, dependent on the assay, the platform, et cetera.
Great, thank you. Do you run two library preps, one for DNA and the other for RNA, and then mix the two libraries? Or do you transform all of the genetic material into DNA and run one single library prep?
So I mean, there are multiple methods out there. I can speak to our particular method, but we've certainly seen other methods in the literature and just through knowledge that do separate out the two. Our particular method co-extracts RNA and DNA, and that's how we built our method. But you know, there's no wrong way to do it, and there's certainly you know, merit to doing it both ways. So it all depends on your validation and how you design your assay.
In live attenuated vaccines, the vaccine virus is present at very high concentrations. Could this dominance outcompete or mask the presence of low-level adventitious agent contaminants in the sample? And if so, what strategies are needed to address this challenge?
Yeah, no, that's a great question, and that's a part exactly the reason why we do a lot of these spiking studies, where we spike in these low, you know, reference viruses that are qualified by you know, labs around the world to and for consistency, right? You're spiking them in at different levels to see what the limit of detection is. And we see a very wide range of limits of detection. I actually had a great slide that I don't have, but Layla showed part of it before. I showed it at the IABS meeting, where it does depend a lot on your matrix. And if you have a large hardy virus of a high concentration, that could essentially suck up, for lack of a better word, all of your NGS reads. Whereas if you have a lower concentration, you can see more of your background. So it's definitely a factor that's assessed in the virus and in the spiking studies that's done for each particular product. But it can be taken further. So there can be method specific or product specific processing steps that you can implement for your particular method in order to potentially reduce some of that that other virus. I mean, targeted capture comes to mind. We've seen presentations on that over the years, and so there are some good methods there. But yeah, that's an excellent point. That's exactly why we do the spiking study, so we can understand what effect the matrix does have on virus detectability.
And Arifa, I see that you've dropped a link to a article within the chat. Did you want to say something more on that article?
Right. So as Brad has mentioned, the spiking studies are useful to inform about the effect of a high titer vaccine or live viral vector virus in terms of the detection. And the one I just put in the chat box, this is a paper that just got published from one of the spiking studies that I highlighted in the list we had, the 2B study, in which we were trying to exactly address that question. But so we did spikes of different levels in the context of a adenovirus, high titer 10 to the 9 virus, but there the background was low cellular background, and we have other studies ongoing in the working group and outside the working group with a more complex background, and also testing long read and short read for detection. And I guess, but the real life samples, I guess, situation in terms of the background that you get, you have to evaluate the background and then put it in the context of virus detection. And I think that's what Brad was trying to present also they test samples that are submitted to them for testing. So I think all of this data is very useful. And another, I guess, aspect could be, is it the type of virus that's in the background? The high titer virus, does that have an effect, DNA versus RNA? So anyway, so I would encourage people to read this paper. Here we did use a DNA virus, so but these are all good questions, and that's the reason you do have to determine the limit of detection for using the reference viruses in the context of your test sample, because it's not just the vaccine virus, but it's also it's more, I think, the host cell material, the nucleic acids that are there that we know do affect virus detection, but the effect of the vaccine virus, may not be, we haven't really, I guess we're still evaluating all of that, yeah. But we do know that in a lot of the live virus vaccines, you could have a lot of host cell background if they're not purified. So that definitely, can influence the detection. So you do need to do some method steps to reduce that.
Yeah.
Thank
Tara,
you, Arifa. Go ahead, Katu.
There is a question that you missed, which may be a general one. Are there any standardized bioinformatic pipeline or common tools for qualitative and quantitative detection of adventitious agent?
So the bioinformatic pipelines, there is no standardized pipeline yet. That's why we're just trying to create materials to standardize the pipeline, such as the database. We're trying to develop data sets so that they can be used to run the pipeline, maybe in silico data sets also to to challenge the pipeline. So unfortunately, there is no one optimized pipeline, but there are papers that have been published, and we're working on some more recent ones that do indicate what considerations you should, I guess, include in your development of a pipeline. We have a paper, I think it was perhaps 2018 from the working group about considerations for optimizing a pipeline, but it doesn't tell you which is the perfect pipeline. It tells you the different criteria that you can consider for development of the pipeline, and then what steps
Yeah.
might influence it.
Great. Thank you, Arifa. And it appears that we have come to the end of the questions. I want to thank Arifa, Colette, Brad, and Layla today for the presentations. I'll reiterate again that these presentations and the recordings will be available on path.org in October. And thank the audience for your attendance today. So this was really meant as a primer for for NGS testing with a specific focus on adventitious agents. I'm going to hand it to Katub now just to give an overview, a brief overview for what you can expect during tomorrow's presentations.
Yeah, thanks, Tara. So tomorrow's presentation is another case study where we are going to be looking at the NGS for molecular consistency in monitoring biopharmaceutical. Here we have Kotsia presenting on that. So today we covered adventitious agent, which is generally for every vaccine testing. Tomorrow we'll have a specific case example, and then we also have, more importantly, a panel discussion that some of the questions that came in the chat related to the pharmacopoeias and regulatory guidance documents, all that can be discussed in the panel discussion tomorrow. So I would like to request everyone to join tomorrow for the webinar. Over. Back to you, Tara.
Great. And with that, thank you again for joining us, and we look forward to continuing the conversation tomorrow. Have a great rest of your day.
Thank you.